Okay, so in this lecture, I want to go through the biosynthesis of the various nucleosides. And I guess we're going to be having phosphates present, so let's call them nucleotides. And I'm going to go through these in the order the book presents them in. I think that was helpful. And what they start with is they start with the synthesis of a important precursor to these, and that's carbamyl phosphate. Phosphate, excuse me. So carbamyl phosphate begins with baking soda or bicarbonate ion. So I guess baking soda has the sodium salt. But anyway, we have CO2 adducting with water. And then what we're going to do is phosphorylate this with ATP. So that's going to introduce a phosphate at the O-. And now this acyl phosphate is going to be hyper-electrophilic at the carbonyl oxygen. And we can add a nice, we can add a nucleophile to it, like NH3, which is ammonia. And that's going to generate our carbamyl phosphate with loss of, I'll just show the PI. I'll show the PI here. So that's going to, we could, oh shoot, sorry. We could have this come up, come down, and then come over. And then we release PI, which is inorganic phosphate, buffered phosphoric acid, so that's PI. And then we form, oh shoot, I got that. Oh no, I'm right. Okay, I'm okay. So this is an interesting mechanism to me because, and I'm going to let NH3 add, and I'm going to lose H plus here, just to keep the mechanism simple. So we're halfway there. Now what we need to do is react this with ATP. This is actually an unstable intermediate that results when ammonia adducts with CO2. Turns out it's not super thermodynamically favorable. And so this is a helpful way to get there using ATP to kind of guide things, accelerate things to get there, because by forming that acyl phosphate, we really speed up the reaction of ammonia with that carbonyl oxygen. Again, if it was just CO2, if you take CO2 and just spread it or add it to ammonia, you could form this same species. But obviously we don't have that much ammonia floating around in cells. Okay, so now at this point, we're going to react with another ATP and release another ADP, and that's going to react at the oxygen atom, and assume that gets buffered to the carboxylate to make an O-, which can then add to ATP to make ADP. And now we have our carbamyl phosphate. Which will be a precursor for uridine, which then gets us into the other pyrimidine nucleotide. Before I go on, though, I want to talk about the source of the NH3. As we're going to see this come up a few times. So the source of the NH3 comes from glutamine. So glutamine is an amino acid. And then we can have an enzymatic hydrolysis where the oh gosh, that's an NH2. Sorry. Okay, NH3 over here plus, oh gosh, minus, get my zwitterion right. Okay, so we're going to take the amide at the end of this chain and convert it into a carboxylic acid. And so that is glutamic acid, or glutamate, as they say, which is another amino acid. I guess I'm changing the zwitterionic form. But anyway, So we're going to get NH3 out by an enzymatic hydrolysis of the amide at the end of our glutamate to make glutamic, at the end of our glutamine to make glutamate, which then releases ammonia, as I've highlighted here. Okay, so that's going to make carbamyl phosphate. So we need that to get kick-started into our biosynthesis of uridine. So biosynthesis of the pyrimidine nucleotide uridine. And we're going to focus on making uridine monophosphate. Which allows us to consider the nucleotide instead of the nucleoside. Okay, so to start, we make carbamyl phosphate. I just showed this, but let's go ahead and just have the overall equation here. We're going to use 2 ATPs to couple bicarbonate with ammonia to make, well, this is going to be a starting material, so let's go down. Carbamyl phosphate. I'm going to abbreviate the phosphates as Ps. Okay, so we have our P, and sometimes it's helpful to put a circle around it. Okay, so that carbamyl phosphate is again an acyl phosphate, so it's highly electrophilic at the carbonyl position. And so what we're going to do is couple that with an amino acid, in this case, the amino acid aspartic acid, or aspartate. And the book uses the this variant. I guess there's just one H. We have this Zwitteran, so we have minus H+. So we've lost an H+ on the CO2 at the end of the chain. But anyway, now this is going to be a good nucleophile at nitrogen once it's deprotonated. I don't really have a lone pair there, so I'm drawing that in blue. But that's going to be my nucleophilic atom. So we're going to have this come over, come back down, lose a phosphate, inorganic phosphate. So I need to lose an H+ here as well to make that neutral for this to work, because as it stands, it's NH3+. Okay, so that's the way that nature usually makes amides, is it has acyl phosphates interact with the amines on amino acids to form an amide. And so in this case, our amide is going to be sort of urea-like. Okay, so there is our amide that results. And this is called carbamyl aspartate. Carbamyl aspartate. So that's not a name, I obviously will ask you to memorize, but hopefully it doesn't make too little of sense. Okay, so now we do a super cool, and I'll talk about this in some detail, but we're going to connect this, this atom with this atom, and we're going to lose water. So this is an enzyme catalyzed condensation, and now our atoms are 1, 2, 3, 4, 5, 6, 6 atoms apart, so we can form a 6-membered ring. And so I'm going to have to do some rotating here to make this all fit. Okay, so if this was this in blue was our new bond that was just formed, then, carboxylic acid here. And so if I number these again, I would have 4 at the alpha position, 5, 6, 3, 2, 1. So now this is written where we have our aspartate, our aspartic acid core here. And so that's a really cool coupling event, because what we have here is an amide nitrogen as opposed to an amine nitrogen adding to a carboxylate. Both of these are poor at their jobs. So this is a cool condensation, because the NH2 is an amide. So that's a poor nucleophile. And of all of the carboxylic acid derivatives, the carboxylate is the worst electrophile among all of the carboxylic acid derivatives. Okay, so how this is accomplished is pretty cool. We use a di-zinc enzyme. So we're going to consider a partial mechanism for the condensation. Okay, so how this works is I'm going to have kind of a stop sign-like geometry. I think the book does a nice job at showing this. So there's my aspartate alpha position. Now we'll go to the beta carboxylate. I have the carbonyl pointed inward. Now over here, I've got my urea, which is a carbonyl flanked by 2 nitrogens, and then hydrogen here, and hydrogen here. Now, so there's my carboxylate, and there's my amide nitrogen that need to pair up together. So, I could just have lone pairs attacking here. But what turns out happens is that we have a basic residue sort of grabbing that hydrogen to push the electron density into the pi system of the aspartate. So of the aspartate carboxylate, we reorganize everything so we're in close proximity. That's a good thing that enzymes can do. But we also have to activate this carboxylate so that it's a good electrophile. And to do that, we use 2 zinc 2+ metals, one of which is also bonding to the base. The other one is bonding to the other parts of the enzyme. And so these 2 zincs activate the carboxylate in a Lewis acid manner. Okay, so that we can have our nucleophile addition. Now, after we do this, we're going to have a 6-membered ring: nitrogen, carbonyl, nitrogen, and this is going to be my alpha CO2 minus, or my CO2 minus at the alpha position, and then we have a CH2, and now we have 2 O minuses after that NH2 adds. We've lost the hydrogen by the base. Now each of these O minuses are going to be coupled to a zinc 2+ ion. And this is going to stabilize everything. Now these zinc 2+ atoms can work together to sort of push off one of the oxygens, and they have this going right to an acidic residue within the active site, probably that one that just deprotonated the nitrogen. I doubt it's lost as ZnO minus. You probably have to have another protonation come in, but either way, we can then lose water here. So I'm going to write H2O lost or minus HO zinc plus. Okay, both of those are fine. Or maybe the oxygen couples between the 2 zinc atoms. There's a few ways we could lose that second oxygen, but that's going to give rise to a carbonyl at the northern carbon. Here, we have CO2 minus, H H. Okay, now this is a cool molecule. This has got a cool name. It's called dihydroorotate. But let's get to the next step. So in the next step, we're going to do a desaturation to remove the 2 hydrogen atoms at these positions. I'm going to use some highlighting here. Let's go to green. Highlight these 2 hydrogens in green. What we're going to use is a flavin nucleotide. I believe it's FM. Yep, it's FMN. So this is the flavin mononucleotide, which then gets converted into flavin mononucleotide H2. We've talked about that before, so I won't go into much detail. So it's going to pull 2 hydrogens off of this system. It's going to start with the alpha to the carbonyl as opposed to the alpha to the carboxylate, but the alpha to the carboxylate will be donated as H- into the FMN. That's going to get us to a molecule called orotate, which is basically uracil, but with this spare CO2 molecule, this CO2 anion sitting around. And there's a good discussion on this desaturating enzyme that pulls these 2 hydrogens off in the book. I'm just not going to go through it. Instead, what I want to focus on is the orotate, the fate of the orotate. Okay, so orotate is actually going to next be coupled with with an enzyme that's going to couple this base to our ribose. And this is going to be catalyzed by orotate phosphoribosyl transferase. And this is just going to attach ribose. Okay, so if you remember when we were catabolizing these things, we needed to cleave the ribose from the base and then break down the base. Now we need to, if we're building these things up, we need to consider how we're going to get the ribose coupled in. I'm just going to write ribose here. We can look at that mechanism. Okay, so to do this reaction, and this is going to be the mechanism for ribose addition, we need to have a good leaving group on our ribose so that we can do a substitution reaction. So to do that, let's consider ribose again. It's not deoxyribose, so from our Hayworth projection, we're going to have 2 OHs down. Now, it turns out for molecular recognition, our CH2 is going to go into an OP. That's a monophosphate. Then we're going to have a down to alpha is the alpha sugar. Don't worry about that. Okay, down to an OPP. So it turns out that if we have a diphosphate, now this whole thing, oxygen included, is a good, I don't need that second arrow, is a good leaving group. OPP. So I show those as circles. Those are 2 diphosphates, or that is a single diphosphate unit, which is a good leaving group. We've talked about that. Okay, so as it is at the anomeric position in the presence of an enzyme, which basically allows for a magnesium +2. That's how diphosphates become good leaving groups, is they interact selectively with magnesium +2. In ATP, we can put the magnesium at the end 2 phosphates or the sort of internal 2 phosphates and activate whether or not you put a diphosphate unit on or a single phosphate unit on your nucleophile. Okay, so here we have the anomeric position, which means we can have these electrons come over and just push off our OPP. That's going to give us what's called an oxocarbenium ion, basically a carbocation that has resonance contribution where the oxygen is positively charged. These are very good leaving groups, or excuse me, electrophiles, and can only be generated when we have a good leaving group like a diphosphate present. But if we pull that off on the bottom within an enzyme active site, we can let the nitrogen of our base come in from the top add here, push the electrons up, and that's going to attach our base. And that is exactly what happens. Now on the orotate, we do have an NH, but the H is being pulled off by a basic residue within the enzyme active site. And so I like to kind of shortcut this until the end. I'm going to draw the ribose just once. Then we're going to go back to what happens to this basic residue, because we're not yet at the at a typical nucleotide or a nucleoside, and I ran out of space. Okay. Focusing on this, NH up here. All right. So we've attached our orotate to our ribose. So orotate plus ribose, that's actually going to give us a nucleotide, which is called Uridine. So Uridine, which is the monophosphate in this case, Uridine is a precursor to cytidine, C of the ATGC group of nucleotides that we need to consider. So all we have to do is lose, we're getting there. We have to do a few things. We need to lose the CO2, and that will give us uridine, the U, I guess, in all of this. And we need to do one more step after that to get to the cytidine. Okay, so I'm going to going forward, I'm going to abbreviate my ribose unit as just ribose or sugar. Okay, so let's get back to the base. So what's happening now? Well, it's a super cool reaction for a lot of reasons, and it's very counterintuitive. But what we're going to do at this stage is lose CO2 and replace it with a hydrogen atom. Now that's not that big of a deal for a lot of in a lot of cases, because you can lose CO2 from things to make negative charges, but those negative charges need to be stabilized somehow. Now it seems like the mechanism for this is decarboxylation in an enzyme active site that weakens that carbon-carbon bond that we need to break, and it weakens it by stabilizing the resulting anion. Now this anion that I'm about to draw is a vinyl anion and is not inherently stabilized by resonance structures. Now I'm not going to go as far as saying it's unstable, but it's not like it's inherently stable. Okay, this isn't a reaction that's that would be great to form on paper without an enzyme. So the enzyme that accomplishes this, the ENZ enzyme, is it has to be uridine. Where's the name of that enzyme? Sorry. Okay. Well, okay. The enzyme provides a rate acceleration. I'm just going to write this. Provides a rate acceleration of 10 to the 17th. So 17 orders of magnitude, which means an uncatalyzed decarboxylation would be 10 to the 17 times slower. All right, I wish I knew the name of that enzyme. I just read it. Shoot, I know it's just in that text. You know how you're talking and you're trying to find what you want to say, but okay. No worries. So we're at an anion. And this anion, again, I can't emphasize enough how cool this decarboxylation reaction is. This, like, nature would not rely on this, but nature got really lucky during the course of evolution to find an enzyme that was capable of pulling off that CO2 minus, obviously critical, otherwise we we probably would have a lot more trouble with a CO2 on that side of the molecule forming our double helix, and then a lot of stuff is going to be an issue if we have CO2 minuses in both our cytidines and our uridines. Okay. Anyway, this just can react with some H+ source to form the base uracil, which being now attached to ribose or ribose 1-phosphate is uridine. So again, I'm not going to worry about the phosphorylation on the ribose right now. Is it a nucleoside or a nucleotide? These aren't the reactions we're focused on right now. Okay. So, well, maybe we should. It looks like the book wants us to at this point. So this is uridine. Now what uridine does is uridine reacts with ATP to form 2 times ATP to make uridine triphosphate. I'm just going to mention this part once because again, I want to do my best to abbreviate the sugar. That's where we get the most sort of productive use of our time. But okay, let's go ahead and discuss the sugar. Maybe everybody's getting really fast at this. I assume it's a little cumbersome to draw. But we're going to let the CO2 now react with 2 more ATPs to convert the uridine monophosphate into uridine triphosphate. Now, it's important to note that during the polymerization of nucleotides to form our strands of DNA and RNA, we need to use the triphosphates of the nucleotides. So let's just mention this. Nucleotide triphosphate is required for RNA, DNA synthesis. So if you ever do PCR where you're trying to you're trying to amplify segments of DNA and actually make, DNA with specific sequences, you're connecting a bunch of A's, T's, G's, and C's to form these chains. So this would be a deoxy version of, well, I have uridine. So we'd want to get to cytidine and then do stuff that way. Anyway, okay, what you do is you add the DNTPs, deoxynucleotide triphosphates, to the reaction mixture, which with your polymerase and your primer and your other stuff, you heat up, you cool down, you anneal, all that stuff, you elongate chains. You're actually using the DNTPs, the triphosphates of the nucleotides, to stitch together to make the growing DNA RNA chain. So again, I want to abbreviate that ribose triphosphate is just ribose, and we don't have to worry about if it's oxygenated or deoxygenated, because the purpose here is to look at the organic chemistry that's happening on the bases. Okay, so we're at uridine triphosphate. What I want to do is abbreviate that to, whoops, I skipped a step here. Let's go here. Some ribose sugar. Now what we're going to do is convert this into cytidine. You may remember that we actually started the catabolism with cytidine and we hydrolyzed it to make uridine, and then we were off to the races for breaking down that pyrimidine. So to get to our other pyrimidine, what we're going to do is treat this with ammonia again. So the first thing is we react this with ATP. This is a common strategy. Now the ATP is going to, well, I'm not going to consider the lone pair as much as I want to. Old habits. We're in a biological system, so we have an enzyme tugging off the hydrogen. It's going to provide electron density into the carbonyl, which is going to push electrons up. We're going to attack the ATP. We're going to pull one of the phosphates off of the ATP to release ADP. That's going to make this structure. Now the monophosphate isn't as good of a leaving group as the diphosphate, but it's still a pretty good leaving group. And now we actually have an aromatic molecule because the nitrogen is going to adopt sp2 character. But anyway, at this point, what we're going to do is an addition elimination process where we have NH3. So here's just NH3. Okay, and we have some basic residue tugging on one of the hydrogens. We're going to have electron density adding to this carbon-nitrogen double bond as an acidic residue is lying close by that basic nitrogen atom. Okay, so the point is when we have this sort of phosphate bonded to a PO bonded to a carbon-nitrogen double bond, we now sort of set that up to act like a carbonyl where we can add and then eliminate. In this case, we're going to eliminate the phosphate as a good leaving group. So what does that look like? Well, we're going to add here, gosh, single bonds already. Okay, we're going to add here to make an NH2 bond, and then we have an OP. Then we can lose the hydrogen to kick off the OP. So it's adding and then it's eliminating, in this case, the OP, and that's going to give us a product where we now have an NH2. Plus a PI, a phosphate that's lost. So when the P has an O with it, it then gets lost as inorganic phosphate. If you are catching up and you really want me to draw that again, I'm happy to. Okay. All right, so alternatively, and I think it's helpful to show this too, I see the book has this written as, if we take the something about this heterocycle, I still am just not super fast at drawing. Let's go ahead and go back to this phosphate. Now, another way that the authors say we could have this react is if we had the nucleophilic electrons out here, push electrons down, and then push over to this oxygen and assume this oxygen is stabilizing. This oxygen with a negative charge is being stabilized by the enzyme active site. And we don't have to invoke an acidic hydrogen in the active site. That gives us probably some more flexibility. This is probably better than what I had drawn. But then that negative charge can swing down and kick off the OP, get back to an aromatic system. Either way, we replace that OP with an NH2 at the top of our heterocycle. If you check your notes, this is the pyrimidine of cytidine. It turns out this happens at the triphosphate level. So we would have the nucleotide triphosphate, cytidine triphosphate being formed because it appears the triphosphate of uridine is what gets converted into cytidine. So we have C and we have U, and those are both of the pyrimidines. Okay. If we think about, if we just take a step back and look at our perspective, what did we have? What did we do? We started with carbamyl phosphate. We then added that to aspartate, did a condensation, then started pulling off hydrogens and a CO2 there. And we're adducting to ribose, and that got us to our pyrimidine system. So it was the aspartate condensing onto the carbamyl phosphate that gave rise to our pyrimidine architecture. And so now we're going to build the purines. So biosynthesis of purines is coming next. Okay, so let's look at the biosynthesis of purines. So this is going to be our, see, we did C and we did U, and we did, didn't we do, don't we have one more? Okay, well, we'll get there. I'm assuming we'll cover all of these. I just want to check my notes. Sorry about this. I know it's easy to fast forward. Oh yeah, so we haven't covered thymine. I wonder when we get to thymine. Just looking through here. Did I miss it? Huh. Well, we'll see. Maybe thymidine is coming. I thought we covered all of them. Okay, so we're going to talk about the biosynthesis of purines. What we're going to do is actually construct these a little bit differently, as opposed to constructing the entire base and then coupling it to the ribose or the deoxyribose. We're going to have the ribose 1-phosphate get converted into the diphosphate at the 1 position. And then we're going to add that to ammonia from the top. Okay, that's a lazy substitution pattern. We lose the OPP first to make an oxocarbenium ion, and then the NH3 adds. But I want to just get to the punchline here as we get started, which is we're going to take this species as our starting material. And this is a ribosyl amine. It's like a hemiaminal or an aminal. I just generally call these things NO-acetals, just to really emphasize the point. But this is going to be our starting material. And we're first going to couple this to glycine. So we're going to couple this to glycine. And to do that, we're going to have the glycine first get phosphorylated. Gets phosphorylated to make an acyl phosphate so that the nitrogen on our ribosylamine can add here, as that will be the electrophilic carbonyl if instead of an OH, that was an O-phosphate. So that's a reaction with ATP. That's going to be an interesting, a nice reaction to talk about during the amino acid section. So I'm just going to hold off on the mechanistic detail there. And it's at this point where it starts to get a little bit more practical to talk about everything happening and just abbreviating the ribose. Okay, so a cool reaction at this point happens, and that is the reaction with tetra 10-4-meal THF, which is tetrahydrofolate. So we're going to talk about that in just a second. Let me get a new page here. And that's going to give us a structure where the amine at the end of the chain, which ultimately came from glycine, now is a formamide. So this is a formamide from THF. Okay. So that's an important step. It's going to happen twice, and we'll look at this in a lot of detail. Well, I mean, we'll just look at it in some detail now, and then we'll get to it. Get on with it. Okay, so 10-4-meal tetrahydrofolate is this structure. So a lot of atoms being added here. Okay. I did do a rotation I didn't want to do, but I wanted to make sure we could clearly see this formamide right here. So this is a formyl group donor. Now what's pretty cool is this actually comes from the tetra. I mean, this is a series of kind of steps related to the tetrahydrofolate cofactor. So what you can do is you can, let's see, how do I abbreviate this without getting too overwhelming? And I feel like I lost a double bond. I did, double bond right here. Well, maybe it's too much to cover, but what happens is these two nitrogens, this ethylene diamine unit, will capture formaldehyde and convert to a formamide. So it would be easy to make formamides, which is just an amide at the end of where the nitrogen, the carbon on the amide has nothing else attached to it other than a hydrogen. It would be great to just make that from formic acid, but formic acid is highly toxic and causes a lot of problems. And in addition to that, if formamide can be a metabolite resulting from some biochemical processes, but we know this, embalming agent is not good for biology. It's very toxic, so we need some way to capture it. And tetrahydrofolate is a really good capture of formamide. Now, what I could do is I could just abbreviate this down to the diamine unit here. So this is the THF diamine. And these R groups, again, we have the rest of our heterocycle as shown by the R group on the left, which I'll write as R prime. And then the R group on the right is just another big part of a molecule. So we'll just keep these two together. I'm going to highlight these as red. These two react with formaldehyde. And they capture the formaldehyde to form an aminal, an NN aminal, where you can think about it as we first. Well, I'll just draw it. I'll just draw it this way. I'll just draw the product. Okay, so we lose water. Here is our diamine system. This dot, this yellow dot, corresponds to the carbon of our formaldehyde. Now, if you look, the formaldehyde is in the same. The formaldehyde carbon is in the same oxidation state. It's not like we did a redox process. It started with 2 hydrogens. It ends with 2 hydrogens. Instead of having 2 bonds to oxygen, now it's 2 bonds to nitrogen. This happens as easily as an imine formation or a Schiff base formation would occur. Okay. So what you can do at this point is you can react this with an NADP plus to form an NADPH, and that's an oxidation event. That's going to remove a hydrogen and oxidize the nitrogen to give us this system, which is now positive here, and then that can react with, I keep forgetting to make a page, sorry. That can react with water spontaneously to make the species used in this biosynthetic process, which has our green formyl group as part of the formamide. Okay, I'm just going to let you copy that down, and I'm going to make a few more pages. Okay. So back to what's happening in this case. What we have is we've basically taken our glycine unit. I'm just going to recap briefly. Taken our glycine unit that's attached to ribose, ribosylamine, and we're going to react the THF. And we call this the 4-meal version of THF. And we're just going to get tetrahydrofolate out, which then needs to capture another formaldehyde, get oxidized again to make more 10-formyl THF. But that's going to transfer, I guess, the formamide from the THF cofactor onto the NH2 sitting at the end of our ring. Okay. At this point, we want to cyclize this so that let's just keep going from here. Okay, so at this point, we're going to react this with ATP. We're going to form an iminophosphate, just like we did before, where an amide now becomes a carbon-nitrogen double bond. Sorry, didn't mean to zoom there. A carbon-nitrogen double bond forms to an OP, which then goes up to an amide. I'm going to start drawing my 4-meal group down this way. And that's now attached to ribose. Okay. What we're going to do is add NH3 to this, and we've seen this before. The NH3 is going to add to the iminophosphate to add, jump down, jump back over, pop off the OP. This is all enzyme mediated, so we can control protonation states as needed. And that's going to give us this species, where we've substituted the OP for an NH. Now it turns out the tautomeric equilibrium is going to convert this enamine core, into a more keto-like core, which is going to be an imine in this case, or a Schiff base. Now, when we are at that point, what we can do is condense our nitrogen onto the oxygen of our formamide. Now, that doesn't happen spontaneously. We need to make yet another iminophosphate where the amide sort of comes down and reacts with ATP to put a phosphate group on the oxygen of our amide. So that's going to look like this now. Okay. Now we can do another addition-elimination process to form a 5-membered ring. So here now the nitrogen is going to add to this position. We push up on the double bond, we push back down, and we kick off the phosphate. If we do that, we connect nitrogen 1, 2, 3, 4, 5. We have a 5-membered ring instead of a 6-membered ring. And so what that looks like is we have carbon-nitrogen double bond, carbon-nitrogen single bond. Then we have our, what looks like our key piece of our imidazole. Now, when we're this close to aromaticity, we are once again going to do a tautomerization to get to a more stable tautomeric form. Here we're going to go from the imine back to the enamine, but we're going to put the double bond inside the ring, because the nitrogen is bonded elsewhere, that's going to give us an aromatic imidazole. NH2 is now sticking out. Ribose is still down here. All right. Now, at this point, we are going to adduct with, so we have a few approaches to this, it looks like. We are ultimately going to adduct with CO2. Now, the book shows a few equilibrium pathways. I just want to directly add to CO2. The way this could look, and again, I'm getting in the habit of just pulling these hydrogens off, is we could have some basic residue come down and then come over. Now, this enamine is going to add to the central carbon of CO2, which I'm going to break organic 1 rules and draw this at an angle, push the electrons from a CO2 double bond onto an oxygen atom. So I really like this step. I really like any step where we can just kind of adduct with CO2. You know what, I'm going to erase, well, I mean, I know a base is involved here, but we ultimately have to lose a hydrogen anyway. I'm going to erase that base, but I'm not going to just do that to my students. I'm going to cross it out. Okay, the reason why I want to do this is this looks like an electrophilic aromatic substitution reaction where we lose aromaticity really quick and then we get it back by just losing a hydrogen atom. So if we do, we could make a carbon-carbon double bond at the position indicated, well, in the final product. Let's just draw the final product out. Form a carbon-carbon double bond and then break the carbon-nitrogen double bond by pushing electrons onto the nitrogen atom. That's going to give back an imidazole structure here. So we had to append another carbon atom, which we did with CO2, a process that I really like. Okay, now it's at this point that we want to get our last 2 carbons on for our ring system. And to do that, though, we have to sort of, I guess, yeah, we're down to the last 2 atoms, a nitrogen and a carbon. So we're going to add the nitrogen first. It's kind of a clever reaction. What we're going to do is, first of all, add this to ATP. And that's going to phosphorylate the oxygen atom with a negative charge to make an acyl phosphate. Then we're going to add in aspartate, which we've done previously. Well, no, let's keep that as NH2. So we're going to come up, come down, and come over. Help if I finish my aspartate. Sorry about that. So there is L-aspartate, the amino acid that's coming in. That's going to make an amide at the position that was the carboxylate, or the CO2 minus that we just added from our CO2 adducting with our enamine. Okay, now an interesting reaction happens where we actually lose a hydrogen at this position and then simultaneously sort of fragment a carbon-nitrogen bond by making a carbon-carbon double bond. So that's a really wonky reaction because you ultimately just convert your carboxylic acid into an amide. So we've added the nitrogen, and you get this weird molecule with two carboxylic acids separated by an alkene. And this is actually just the molecule fumarate, which we've looked at before in other metabolic pathways. Okay. Ribose, ribose. A lot to draw with each of these steps. You're probably like, yes, Willoughby, I get it. Okay. Sorry. All right, so we're almost close to adding our atoms of our purine system. We just need one more carbon to bridge those two NH2s. So what we're going to do is just add some more 10,4-methyl THF. And that's going to put a converted NH2 into a formamide. So in this case, it's going to be the NH2 that's not part of the amide already. So what that looks like is we have our 5-membered ring. We don't yet quite have our 6-membered ring, but we are close. Still have an NH2 up here. We now have an NH2, a formyl group. So there is our formamide. Okay, so at the very least, we've added the carbon necessary to form the formamide. Now, all we have to do is form a Schiff base or an imine from this nitrogen to this carbon. Now, if we count out how far away those are, 1, 2, 3, 4, 5, 6 atoms away, so they'd actually form a Schiff base across a 6-membered ring. So again, Schiff base is going to be the imine formation across those two atoms, and then we have our 5 to 6-membered ring system. Because we've connected these two positions now by way of a Schiff base. Okay, so we have Schiff base formation, and then that's followed by a tautomerization, where we just flip the position of the double bond in the hydrogen in this system. So we'll get a tautomer of this. But to those of you that have these things memorized, and the reason why this tautomer is favored is probably something to do with the upper nitrogen being an amide. But to those of you that have all of the nucleotide structures memorized, you're probably thinking to yourself, this doesn't look like a purine nucleotide that I've seen. And that is indeed the case. This is inosine. And so inosine sets the stage for the formation of our adenosine and our guanosine. So what we can do with these, what we can do with inosine, and again, this is our common starting material for A and G, is we can take this and we can take it under two pathways. So divergent pathway, maybe I'll try to get ambitious and show both of them. Okay. So if we take this structure and we do an and this is GTP, we could do GTP and aspartate, and we get GDP and PI. That's going to convert this amide, oxygen, into an iminophosphate, which we can then adduct with aspartate to form what's called adenoyl succinate, and it's very close to adenosine. Okay, so that nitrogen that I just wrote is the nitrogen of aspartate. Okay. And what we can do is actually just lose, once again, we can just lose fumarate from this to give us our NH2. So this is now adenosine. So there's our A. Okay, we're going to do a similar series of events to get to guanosine, but we're going to start with an oxidation. So this is going to be NAD++ water is going to accomplish an oxidation at this position that I show with a star. So we're going to redraw the base, but we're going to have a hydroxyl group at that position. And that hydroxyl group will tautomerize to a carbonyl. And then with that carbonyl, we're going to do the same reaction that we've done a few times now, where we convert that to an iminophosphate. Sneak by adenosine here. So we're going to react this with ATP and NH3, which is from glutamine. But we're going to substitute the highlighted oxygen with an NH2 by way of this iminophosphate, where we have the oxygen now bonded to a single phosphate. It becomes a good leaving group for additional elimination sequences. We've done this a few times. I don't want to get too redundant because we've already talked about so much. But that's going to introduce our carbon-nitrogen double bond and then put an NH2 at this position. And so this is now guanosine. Okay, so a lot of the steps are sort of redundant. And so at this point, I don't think I'm going to, the oxidation to that hydroxyl group is really interesting. But at this point, this lecture is feeling pretty long already. So I'm going to stop here, and then we'll do one more lecture on the deoxygenation of ribose to get to the deoxynucleotides, and we'll call it a chapter. Okay? So with that, I'll end here.